I'm Jim Collins. I'm a bioengineering professor at MIT, Harvard's Wyss Institute, and the Broad Institute of MIT and Harvard. And our group is largely working on synthetic biology, engineering biology to create new classes of diagnostics and therapeutics. In 1989, when the Berlin Wall fell, I was a graduate student at the University of Oxford. So our research on a wearable face mask really addresses the challenge of how can we harness the power and diversity of biology, and more specifically synthetic biology, without utilizing living cells. We recognized a few years ago that you can take cell-free extracts, that is the machinery of a living cell outside of a living cell, and freeze dry it onto paper and other materials, including clothing. This machinery that would include DNA, RNA, ribosomes, other molecular machines, nucleotides, ATP, and other biomolecules can be coupled with synthetic biology components, for example, sensors that can detect pathogen RNA or pathogen DNA, and now create portable, easy-to-use diagnostics. We initially did this effort to address the Ebola and Zika crisis in 2014 and 2016, creating a series of paper-based diagnostics. In the case of Zika, these paper-based diagnostics were actually used in 6 different countries during the 2016 outbreak for both research and surveillance. On the heels of these efforts, we recognized that we could extend these paper-based efforts to wearable diagnostics, where the idea would be you could have patches or forms of clothing that could be used to report out, have you been exposed to a pathogen and/or are you infected? And our idea was that we could use these to address challenges for healthcare workers, first responders, and military personnel. We were extending this work really to create things such as the lab coat of the future when the pandemic hit. And our team here at the Wyss Institute at MIT recognized we had an opportunity to use this platform to address the challenge of can we get after wearable, rapid, inexpensive diagnostics to address the COVID-19 pandemic. And our idea was to create a small wearable insert that could be added to any face mask. The notion would be that during the normal act of coughing, sneezing, breathing, you'll give off vapors. These vapors, if you're infected, will include the viral particles of COVID-19, SARS-CoV-2. And our system was then set up to collect those, expose the viral RNA, amplify it, detect it with CRISPR elements, and report out whether you've been infected. And we showed this wearable component could report out in a matter of minutes and be highly specific to SARS-CoV-2 relative to other coronaviruses. Since initially introduced this, we've now shown you can multiplex this system and actually have a report out on whether you are also infected or infected with the seasonal flu. So we're really excited about how this platform has enabled us to move the power of synthetic biology out of a laboratory into our communities in the form of this wearable face mask. Many challenges remain for our face mask diagnostic. I think most notably, can we manufacture the device inexpensively so that all who need it can obtain it? I think what keeps me up most at night is can we appropriately cross the valley of death and move our innovations out of our academic lab here at the Wyss Institute and MIT into the commercial space. My outlook for the future is really one of optimism. I think we've seen tremendous advances from synthetic biology during the pandemic, and I think synthetic biology will enable us to make biology and medicine more programmable, expanding our capacity to address newly emerging pathogens and better prepare us for the next pandemic.